This video will discuss respiratory enzymes, which are vital for cellular energy production. We will cover their history, the major stages of respiration, and their overall importance.
Here is an outline of the content we will cover: the introduction, history, major stages of respiration, cellular respiration, what respiratory enzymes are, their importance, and a conclusion. Respiratory enzymes are specialized proteins essential for energy production. They catalyze the breakdown of nutrients like glucose, fats, and proteins to produce ATP, and are highly specific to the reactions they catalyze. In the 19th century, Louis Pasteur linked enzymes to life processes. Key discoveries include Edward Buchner's isolation of zymase in 1897 and the identification of coenzymes in the mid-20th century. In 1937, Hans Krebs discovered the key enzymes of the Krebs cycle. The major stages of respiration include glycolysis, the Krebs cycle, the electron transport chain, and ATP formation. Cellular respiration is the process of breaking down glucose and oxygen into carbon dioxide, water, and energy, or ATP. It occurs in all living cells and is essential for energy supply and life processes. The main stages are glycolysis, the link reaction, the Krebs cycle, and the etc. Respiratory enzymes are biological catalysts that significantly accelerate metabolic reactions, especially in the step-by-step breakdown of glucose. Without them, energy release would be far too slow. This slide shows a visual representation related to respiratory processes. Glycolysis breaks down one glucose molecule into two pyruvate molecules. This anaerobic process occurs in the cytoplasm, producing ATP and electron carriers like NADH and providing intermediates for other pathways. It happens in three phases: energy investment, cleavage, and energy payoff. This slide shows a visual representation related to respiratory processes. The energy investment phase activates glucose using ATP. Key enzymes include hexokinase and PHOS-PHO-FRUCTOKINASE1, which is a major regulatory step. The cleavage phase then splits the 6-carbon glucose into two 3-carbon molecules. The energy payoff phase involves reactions where NADH is produced, capturing high-energy electrons. ATP is also produced through substrate-level phosphorylation, ultimately converting the molecules into pyruvate using pyruvate kinase. The Krebs cycle, also known as the citric acid or TCA cycle, is an aerobic process in the mitochondrial matrix. It completes the oxidation of glucose, starting with acetyl-CoA to produce ATP, NADH, FADH2, and CO2. The cycle begins with the formation of citrate and isomerization, catalyzed by enzymes like citrate synthase. The oxidation and decarboxylation steps follow, producing NADH, CO2, and ATP or GTP, using enzymes like isocitrate dehydrogenase. In the regeneration of oxaloacetate phase, succinate is converted back to oxaloacetate through steps that produce FADH2 and NADH. The overall yield per acetyl-CoA is 3 NADH, 1 FADH2, 1 GTP, and 2 CO2. The electron transport chain, or etc., occurs in the inner mitochondrial membrane. It involves complexes that transfer electrons from NADH and FADH2 to coenzyme Q, pumping protons across the membrane. Oxygen is the final electron acceptor. This figure illustrates the mitochondrial respiratory chain complexes involved in the etc. ATP synthase is a protein in the mitochondrial inner membrane that functions like a molecular motor. It harnesses the energy of protons flowing down their electrochemical gradient to synthesize ATP from ADP and phosphate. The process involves building a proton gradient, followed by proton entry into the F0 subunit, causing the C ring and gamma stalk to rotate. This rotation drives conformational changes in the beta subunits, leading to ATP formation and release. This figure shows the structure and function of ATP synthase. Respiratory enzymes are important because they produce ATP, speed up respiration, control metabolic reactions, drive electron transport, build the proton gradient, and support ATP synthesis. They ensure efficient oxygen use and maintain cell energy, growth, and tissue repair. Defects in these enzymes can reduce ATP production, leading to muscle weakness, fatigue, and mitochondrial myopathies, such as Lee's syndrome. Problems in etc. complexes cause energy flow disruption, resulting in lactic acidosis and failure of high-energy demand cells like brain and nerve cells. In conclusion, respiratory enzymes are vital for cellular respiration, facilitating electron transfer and energy generation. Key examples include cytochrome C oxidase, succinate dehydrogenase, and NADH dehydrogenase. This slide provides space for acknowledgements or further resources.